we have 5 major areas that we want to look at today. The first one would be methods to determine the analytical threshold. We'll go into some background on this. There are a variety of different ways of establishing analytical threshold. And more recently, with the sensitivity of the PCR kits and the CE instruments, improvements in background noise have been made, and so analytical thresholds in general are lower than they used to be. So we'll look at determining analytical threshold. Also, we'll look at using allele-specific stutter filters. This is something that GeneMarker HID has had in place since about 2016, I believe, and it's extremely helpful in looking at complex mixtures, and also to provide the best information for your PG mixture analysis. After looking at allele-specific stutter filters, we'll look at establishing and using a contamination database, and then other things to consider, looking at control concordances, analysis templates, the different types of reports that are required, and also locking in a validated SOP to avoid any accidental deviation from the process. And lastly, we'll end up with something, the audit trail, which of course is essential to be saved with the projects. So we'll starting with methods to determine analytical threshold. We went, of course, to the OSAC guidelines on analytical and stochastic thresholds. And so this is just a starting point. Everybody's familiar with what an allele peak is, the analytical threshold. This is something that varies from lab to lab, instrument to instrument, and even on the same instrument, depending on the CE conditions. Artifacts, these are amplification of non-targeted DNA template. And so they're recognized products of PCR that are not allelic peaks. And then the noise. These are meaningless output that occurs on all the electronic equipment. It's random. It's generated during the CE separation, and establishing an analytical threshold is essential to determine at what point above what RFU is there high confidence that this is an allelic peak and not noise. All right, so when we looked into the OSAC guidelines, there are many acceptable published methods for determining analytical threshold. The OSAC guidelines in the appendix actually list 9 references. So there are many published references. Dr. Catherine Grigacek detailed 5 methods in a workshop years back with a section module 3 on setting analytical thresholds. 3 of these methods use negative controls, and they're typically lower. The results are typically lower. 2 of the methods use samples with amplicons, and she makes excellent points during this presentation that consideration should be given to what type of data will be analyzed, and recognizing that if there's more DNA in a sample, then the baseline can also be higher. One of the other things that labs will often do is change the injection time, and that will impact the analytical threshold as well. One thing to note is that a common method that used to be used is no longer recommended by OSAC, and that is the method of looking at extreme value calculations, where you're looking at the highest noise peak and the lowest trough, and then multiplying that difference by 2. This is actually one of the methods that used to be used fairly commonly, but is no longer recommended. Okay, let's take a look then at a common method that is used in many labs for determining analytical threshold. And it goes through these 7 steps, which you're familiar with: amplifying the samples per the lab's SOP, and then collecting the FSA files from the capillary electrophoresis instrument, analyzing the FSA files at 1 RFU, so no y-axis requirements, no filters, just call everything. Then the analyst needs to go through each sample one at a time and delete all the actual amplicons, the stutter, diblobs, any of the known artifacts, things that are artifacts, so that only the noise call remains. That table is then exported, and typically in Excel or spreadsheet, you calculate the average noise and the standard deviation for each die. I put a little note in here because some labs use dye-specific analytical thresholds because of differences between the noise in the different dyes. Alternatively, OSAC does state that you can employ a single global analytical threshold for all dyes as long as you provide statistical support that it's not significantly different. The noise level is not significantly different from one dye channel to another. Okay, and then the analytical threshold is the average plus 3 times the standard deviation. And I always knew that 3 was a commonly used number, but what I didn't realize was it's per Kaiser that a K of 3 when the noise is normally distributed, which is common, is 99.86% confidence. So 3 is a commonly used K value for this calculation. All right, so continuing with this method, step 4 in that previous slide is very time consuming. It has potential for error if artifacts are missed. And so we wanted to take a look at a software tool that can assist with this. And this is GMarker HID version 3.0 software. And we have there's a validation assistance tool here for analytical threshold. And so when this is selected, a pop-up box comes through, and we have selections for the type of baseline subtraction and pull-up correction smoothing and spike removal. So we'd say okay to that. And the screen then that is displayed is the analytical threshold tool. It's automatically detecting the peaks and artifacts, and it does not call the allele peaks, the stutter peaks, the areas of pull down. These are not detected, and so we have then a noise table. So if we look at the left side of that screen, we're looking at the noise peaks, and you can review each sample. I've just pulled up the blue dye on one as a visual here, that it's calling it's calling only the noise peaks. Here there was some pull-up peaks from the size standard, and it's done for each sample, each dye. And if I go to the next slide, the other tab one here is the limit of detection table. And this table is organized across the top with the total average peak height, standard deviation, total LOD. So if we were not differentiating between the dyes, we'd have an average peak height very low, standard deviation is quite high, and then the average plus 3 times the standard deviation, so an analytical threshold across the dyes of 21 to 22. But it also is arranged so we have the average peak height of the blue channel, standard deviation, and then the limited detection in blue. And this is what we see here is quite common. The blue tends to be lower noise. The green is higher. It's unusual actually in this one. The yellow, in my experience, has typically also been high. It's lower here. And the purple up here at 22.3. So with the LOD table, the user selects the K value. Up here, the software provides the peak height averages and the standard deviation, and the LOD is here in the last row called all samples. So this is something to much more quickly done than manually going through and removing the peaks and the stutter peaks. And also it's following a set of rules, and so there's not the potential that we would miss, that an analyst would miss deleting a peak or stutter peak. The software recognizes that and provides only the noise peaks in this table. Once the analytical threshold is determined per dye, in gene marker HID, you would add that to the marker. And so we'd have the homozygous intensity is set here in this example just at 50. And this inconclusive range is a stochastic setting. So if your stochastic is below 100, this would show that peaks that are above 50 but below 100, when there's only one peak in the sample, will be flagged as inconclusive to alert you that it's in that stochastic range. Incorporating the dye outside the marker, this is just by dye in the analysis template. You can pick the percent filter and the minimum RFU per dye. Okay, so now we're ready to go to the second topic, which is using allele-specific stutter filters. And as I mentioned, the marker-specific stutters were the most commonly method for filtering out stutter, but with the complexity of markers, we found that allele-specific or range-specific, alleles 7, 8, and 9, and then 10, 11, 12 might have different ranges. And this has become especially helpful for samples that are going to later be analyzed for mixture analysis. Okay, so let's look at some background. Traditionally, the average for the marker plus 3 times the standard deviation. And the only issue here is it's not accommodating variation within the marker. And also typically, marker-specific stutters typically not additive. So 2 shortcomings there of the traditional method. Stutter variation can occur from any number of reasons: the length of the repeat, the complexity of the repeat, size of the marker, size range of the marker, and the position if it's an additive effect of stutter, which is extremely prevalent in mixture samples. Okay, so there are different outcomes. If the stutter filter is too high, you may miss contributor peaks. Actual contributor peaks would be potentially filtered out. And if the stutter filter is too low, it increases the number of analyst interventions or the potential to include an artifact peak into your mixture. So this is just background information. And this slide we included just because it shows a variety of stutter. Here we see the -1 repeat stutter, the shared repeat stutter, some complex stutter down here. So this is just showing a typical single source sample with plus stutter, minus stutter, half repeat stutter, and very typical for an STR profile. And looking at them, just at the difference here, the impact of additive stutter, just 2 samples within the same sample, that the peak in that additive position, it's got -X stutter from the 11 and +X stutter from the 9s, where X is the repeat number. So this is very commonly seen that very different peak heights, and it's because of that additive effect of the stutter position. It's a shared position. Okay, looking at the length of the repeat, trinucleotides, shorter repeats typically have much greater stutter. And the stutter is very pronounced here, even in the +1 repeat position. Whereas a pentanucleotide, much lower stutter, virtually no. Maybe there's a tiny little blip here at the plus repeat. So the length of the repeat impacts the stutter. And the complexity of the repeat. A homogeneous repeat is typically very linear and mainly has stutter at the -1 position. But a heterogeneous repeat, something like SE33, where there is a complex repeat pattern, it's going to have -2 base pair stutter, -1 repeat, or 4 base pair stutter. Here it is with +2 base pair stutter. And so a heterogeneous marker is going to have much more complex stutter patterns. And then also the size of the peaks and the size range of the marker. So the larger the repeat will have different stutter than one within the same marker. Penta E, SE33, some of these markers that have very wide size range will show differences within the marker on the stutter values. So how do we determine that? And here what we did was we relied on a presentation by Dr. John Scheinman and his colleagues in Connecticut State Lab. They presented this work at one of the ISHE conferences. And he has good detail on how they determine the allele-specific stutter values. And so just looking at this sample, looking at one locus here that has a very simple n-1 repeat stutter pattern. And you can see it's nice and linear. And he just explains what they did was looking at the 13, which was the most prevalent. They took the average here and they calculated the slope. And then the other values were set based on this standard curve. Looking at another locus, D1 has a bimodal distribution, so a more complex repeat pattern, and it's based on the longest uninterrupted stretch. The stutters basically correlating to that loss. And so here we can see the whole repeats, and here we can see the ones that have the 0.3. And this is also seen in the table here in terms of the averages. And they followed basically the same process, but they did one for the whole repeats and one for the 0.3 repeat, or the 3 nucleotide repeats. So implementing this as an example in gene marker HID, this is the homogeneous repeat example. And so taking the tables from their calculations, 8, 9, and 10 were at 8.4, 11 was at 9.5, 12, and so it increased as we got larger. So a nice linear stutter pattern. There was not significant plus stutter or -2 base pair stutter or +2 base pair stutter. So these are the markers everyone loves. So the nice homogeneous repeat with a typical stutter range. But even with this typical stutter range, it's ranging from 8.4 all the way up to 17.9. So an average and standard deviation would have the potential to miss real peaks or call artifact peaks. Implementing the other example where there was a bimodal distribution, and it's a little harder to see because it's a larger table, but basically it's going the whole repeat, and then we've got the 0.3 as at a different, and the whole repeat and the 0.3. The 9.1, 0.2 were included because depending on the location of the allele, you needed to have those for the 0.3 of something that was at a position other than an 11, 12, 13, other than a whole repeat value. Okay, so when they looked at this, the n- repeat ranges had significant stutter differences due to size and loss. The n+ and the n-2 base pair or half x were consistent. And so in that n+x and n-half x, the value was the same for all alleles. It differed only in the whole stutter in terms of the n-x position or n-1 repeat. And the benefits of this are also summarized in their presentation. This is still from Scheinman et al. And what they found was significant improvements in their ability to filter the stutter and not filter actual allele peaks in their mixtures. And here in this table, it's showing that they did they did 82 person mixtures, and allele-specific stutter threshold failed to filter 4 stutter peaks. So the allele-specific failed to filter 4 peaks, but the locus-specific threshold actually filtered out 7 real peaks and did not filter out 23 stutter peaks. So the allele-specific stutter was a big improvement over the locus-specific stutter in their 2-person mixtures. And then they looked at 43-person mixtures, and the allele-specific stutter thresholds there did fail to filter 5 stutter peaks, so they were included, and it filtered 2 actual allele peaks. But the locus-specific stutter filtered 13 allele peaks and did not filter out 38 stutter peaks. And so he has here an image. The stacked algorithm is what is helping here, catching that additive effect of the shared stutter position. So then just a summary of the allele-specific stutter filters. Given that n-1 repeat stutter percentage increases with allele size and the use of a single allele threshold, then it's just not optimal for looking at the locus with a single stutter threshold. And with previous analysis software options, we can only have single locus stutter threshold. The filter would either under or overcompensate and require a lot of manual edits. And so this was their summary as to why they switched over to using the allele-specific stutter in the GMark HID software. Again, the advantage is it's allowing for shared position and additive effect of stutter from different alleles. It uses the value of the allele or the allele range. As we saw in the example, sometimes going from one allele to the next, there's a difference. Other times there were 3 or more alleles in a row that had the same allele, same stutter range. It decreased for them the number of analyst interventions and missed fewer actual alleles. Now, how would you test this? Okay, here's just a short example of how you would test allele-specific stutter and the additive stutter. And in this example, we've got a 13 and a 15 allele, and the 14 is in the shared position. So 13 we're looking at n plus x from 13, and we're looking at n minus x from allele 15. And so when the RFU from stutter, we looked at, okay, how much stutter, if the stutter filter is set at 0, then of course we'd expect 0 RFU from the stutter. And for allele 15, if the percentage is 8, we take the percentage here of the minus x stutter, and that would result in 903.36 RFU could be the minus stutter RFU for the 14 allele. And the setting then in the software is to look at the 15 allele. We're looking at the minus X stutter. We're setting it at 8%. 8% would be 903, which is just under this 10,020 RFU for this peak. And it's called because it's above the stutter setting. Okay. So now we want to check, though, then the additive effect of the 13. So I'm going to go to the next slide. And here, if we just have 1%, so from the 13 allele, the amount of RFU from the 13 contributed to that 14 allele, when this is set at 1%, would be 117 RFU. Keeping the 8 the same, that would bring the amount of RFU from stutter to 1020.39. And if you remember, the peak height was actually 1020. Okay. So we'd go into the settings. We'd look at the 13 allele. The plus stutter position is set to 1%. keeping the 15 at the N minus position at 8%, and the peak is filtered out. And so the reason this is a straightforward, simple test, just looking at the N plus stutter. And so for the allele specific stutter values, is looking across the board at the allele setting for each position and whether or not they're in a shared position. It's additive. Okay, when using allele specific stutter filters, one of the ones that I checked was D10. And this is an example of marker specific stutter. I used a sample from the Proved It database. It was a Fusion 6C, and I had 165 alleles. The mean stutter was about 8.9%. The standard deviation, 0.022. And here, interestingly, the maximum stutter peak, and I was looking at anything that was not shared here. The maximum allele stutter peak was 15.5%. The minimum was only 5.3%. And so the mean, though, was the mean plus 3 times standard deviation was up 15.74%. So even more than the maximum. And so this would potentially miss minor contributor peaks if I was using this traditional average plus 3 times the standard deviation. Okay. Okay, so that brings us to the third category, which is establishing and using a contamination database. This is a very short portion of the presentation, but we included it because it's an important process in the forensic labs that with the increased sensitivity of STR kits and the instruments, we're able to detect lower and lower amounts of DNA. And so the potential of detecting the DNA of someone who handled the evidence is actually increasing just because of the sensitivity of the methods. And so typically the genotyping step is the initial place to flag a situation where a casework sample may have a contaminant from your elimination or your contamination database. The second place to flag this would be during your PG analysis, but the CE step is the initial place to detect contamination. So examples of individuals who should be considered for inclusion in the database: crime scene investigators, the sample collection team, the staff of the DNA laboratory. I was in a lab at in Eastern Europe. Their elimination database included anyone who came in the building. This database may be large or small depending on the size of the institution, and it needs to be updated anytime there's a change to staffing or duties of the staff. And so establishing a contamination database, including some screenshots from GMarker HID software, the samples are genotyped, and only the ones that have a clean genotype are included here. It has the ability to put information about that individual here, and then it has the genotype that would scroll over for the rest of the genotype, and that could be submitted to the database. If there's an existing database, if it's in a CMF file from a CODIS compatible file, that can be brought into the database, or it can be a formatted text file. So 3 different ways to get genotypes into the contamination database. When establishing a contamination database, we'll take a look here. Here is a mixture, and it's selected here. It's sample. It's a 3, 4, 5 person mixture. So we can see it's quite complex. It's got some minor peaks to it. And when we go to the contamination check and we check it against the database, we find a positive comparison here and a high comparison here. So this is looking at the database. And so it's detected different samples that are potential contaminants in that. I didn't put a screenshot within the project, but you can also look within the project for any bleed through or crosstalk, depending on the instrument, from sample to sample within the project. This is an example, though, of looking at the contamination database, which would be your elimination database. And we look here at their different settings up here, because especially with mixtures, you may want to limit some of the considerations here, making sure you're looking within your contamination database and not the relationship testing database. And it can also highlight, if there are a large number of samples here, it can group and highlight them. And again, there are ways to go directly to the database to save to that database or search it. Okay, so again, a very short section on contamination database, but an essential part of developing the lab's SOP and validating processes. Okay, so then we have another group, which is more of a catch-all. It's other things to consider. And we're talking here about control concordances, analysis templates, reports, and ensuring that the templates are protected from accidental changes. So in looking at this, we've divided it into 3 sections. We're going to introduce things at monitoring the controls. There are positive controls, negative controls. Some chemistries have internal control probes. So this is our whole group here. All these are considered as control monitoring, analysis settings, and reports. Then we'll look at user access rights and standardizing reports, and then implementing it. So evaluating positive and negative controls. Positive controls, there are typically amplification positives that are supplied with the chemistry. So those would be like 2800M, which we see a lot in here, NIST-C. So a standard DNA that's available to everybody, and the genotype from that particular kit. But many labs will have extraction positives, and those will vary from lab to lab or from time to time within the same lab. And so there is the ability in gene marker software to have 2 different positive control genotypes. And so this software, when it does a control concordance, is going to look for the genotype associated with positive 1 and the genotype associated with positive 2. A negative sample, these would be any samples that you do not expect to have amplicons in. So a no DNA, a reagent blank, a water blank, just the naming, if a naming convention is used, they could all be automatically recognized by the software as a negative. Okay, so now we're going to go to internal control probes. Okay, these are control probes that are included with the PCR kit, and it's to help to evaluate the PCR reaction and the CE separation. And so in this situation, these are peaks that would be successful as long as you have both of them amplified. And so this is a table that we would see the analyst with these internal control probes using. And so if PCR is successful, but there's absence of DNA, there will be both of the internal control probes will be a similar height, but there won't be any sample peaks, no allele peaks from your sample. And without the quality control fragments, you aren't sure, is it because there wasn't enough DNA, or is it because of PCR failure? So here, as long as there are these 2 peaks and there are similar heights, they are, we know that PCR amplification was all right, but there was an absence of template DNA of the sample. If, on the other hand, you lack either quality fragment 1 or quality fragment 2, and no samples appear in the peak, then points towards PCR amplification failure. Inhibited DNA would result in a low molecular weight control peak of being normal height and the higher molecular weight peak of a decreased height. And so that would point towards there's some kind of inhibitor of PCR in the sample that's being amplified. And a fourth condition, if the quality control fragments appear to be similar height, but the sample shows a downward trend, a slope of decreasing height in the larger loci, significant decreasing height, not the typical, yes, it's a larger fragment, and so it's not amplified as many times. That points towards degradation of the template DNA. So this is something that is included, these internal control probes. I think all the manufacturers of kits have an option for including internal control probes in some of their chemistries. And so this is a kind of a rubric that actually it was a few years ago. Someone in Texas did a presentation where they showed that they greatly enhanced the efficiency of their evaluation of what was the problem by using those internal control probes. And so it helps them to differentiate between, was it not enough DNA? Was it a failed amplification? Was it an inhibitor, or was it degradation? Okay, so in GeneMarker HID, there's an automated process for internal control probe evaluation, and it's a QS flag in the electropherogram. So if I look over here in the electropherogram, these are some test samples that all had some issue with them. And when you mouse over that QS, it tells you that Q/S/Q imbalance. You see that there's a nice tall peak here at the low molecular weight in the sample. There are no sample peaks, no amplicons, but there's also not that high molecular weight internal control probe. And so this points towards PCR inhibition. The second one on the list is showing that the Q allele is missing. Both of them are missing. So this points towards amplification failure. And so it's evaluating it via that rubric that we saw on the first slide, and then giving you the feedback here when you mouse over it. In this third example, the Q fragment is quite high, and the S fragment is nicely balanced with it. Okay, so there's nothing inhibiting the PCR process, but when we look at the amplicons, there's decreasing amount, significant, strong decrease here. And this is pointing towards more of an issue of the samples degraded. So there wasn't a problem with the PCR reaction, but more an issue of a degraded template DNA. And here it's giving the option to make your setting to detect at what slope you want to flag it as degradation. When we look at the overall screen, that was a summary of it. The QS red flag appears over in the file name tree, and it also appears in this report over at the left, which would be what would be exported to the lab's LIMS system. So it could be, if it didn't get captured here by the analyst, when it went into the LIMS system, it would alert them that there was a quality problem with their samples and the internal control probes. Also of note is that on a negative control, something that the software is told, yes, this is a negative control, we don't expect to see peaks in the amplicons, but we do expect to see the Q and the S. And so in these situations where the quality internal control probes are used, the negative control will not fail just because it's detecting these quality fragment peaks. Okay. All right, and now we're ready to just touch on that topic of ensuring that the analysis templates are not altered. I think most labs will have access rights, and this is an example specifically of how to prevent a template from being altered accidentally, and that's through user management and access rights. So a typical analyst would have the ability to select one of the templates. But if you look over here, this is all grayed out. They cannot change the panel. They can't change the size standard or any of the settings within the template. They can go to another template, which is validated, but they cannot accidentally change those analysis settings. So that would be in the analysis template. Another place where labs sometimes have reported issues is with just reporting. So here with the CODIS compatible reporting, automatically it's going to only check the ones that had no yellow, no quality flags. Has nice, easy drop-down menu for selection and filling this out, and then save a CODIS compatible report. But the setting is that it only accepts those that did not have any quality issues. So these two samples, the red question mark, there was some quality flag triggered, and so those are not exported to the CODIS. The LIMS report settings, this is another place where labs mentioned to us that sometimes people would accidentally change the report setting and then the LIMS system didn't know what to do with it. So there is a separate LIMS report that the admin is able to establish the LIMS report settings, picking from any of these columns and any of these options. They set that up for their lab, and then the LIMS report is established, and only users with access permissions can change it. So that's also a way to ensure smooth, consistent reporting. You're just showing that the admin then removes the LIMS report setting box. So now when they click the save report, save the LIMS report, it goes directly to, do you want to save it as a text file, a CSV? The typical user would not have the ability to change that. They can just say, yes, I want to save it. It's going to go directly to the location that is mapped on the software for reports to be saved to. Now, the last section of the genotyping portion of the workshop is the audit trail. Okay, so this is, of course, an essential part. The only thing that the administration needs to do is to set up user management so that it will record the individual's ID when they make the report. Okay. So even if they don't set up the ID of the person or the individual users, whenever an edit is made, in this example, a -A peak was deleted, and then the analyst can go to edit comments. And these are just some example comments. It's when you first open the software, it's blank. The lab puts in their list, it can grow. And that way, any of the routinely used comments, you would just select from here, say okay, and that adds the comment then to this cell of the table. And this table is saved with each project and can be, of course, exported with the electropherograms or the report tables. Looking at the audit trail for an entire project, here's the part where it said you have to have user management activated. So it has the analyst's ID here, tells you the project, the version of the software, and this institution name is also entered with user management. And this is just showing it's actually a 38-page audit trail. It was an extensive project that has this trail here. It was edited, and this can be saved as a PDF, or if you want to just visualize it in the software, it's available through a drop-down box in the user interface.